We're going to continue with 7.2 and we're going to look at the general process of transcription first. So your general understanding is that transcription occurs in a 5-to-3 direction. So this is no different as to how you saw DNA replicate, which also replicates in a 5-to-3 direction. So in DNA replication, we saw how DNA polymerase can only add in a 5-to-3 direction. In transcription, we're going to see how RNA polymerase can also only add in a 5-to-3 direction. So transcription is the process by which a DNA sequence or gene is copied into a complementary RNA sequence by RNA polymerase. So here we see RNA polymerase bringing the nucleotide, and the nucleotide matches the corresponding base. And notice that every time that the nucleotide is added, notice how 2 phosphates are released. That's because you require energy, right? So RNA polymerase covalently binds the free NTPs or nucleotides in a reaction that involves the 2 additional release of phosphates. So again, the 5' phosphate is linked to the 3' end of the growing mRNA strands. Okay, so here we see transcription occurring. So remember that transcription happens in the nucleus, and remember that mRNA and tRNA and rRNA are all synthesized through transcription. Also remember from HL1 material that RNA is single-stranded. All right, so the process of transcription can be divided into 3 main steps: initiation, elongation, and termination. So what it's important for you to remember is some of these terminologies and vocab words that you probably didn't learn last year. So in the first step, known as initiation, RNA polymerase binds to a promoter. So there's got to be a promoter region within the DNA strand. And this causes the DNA strand to unwind and separate itself. Then elongation occurs, and here's where RNA polymerase moves along the coding sequence, synthesizing in a 5' to 3' direction. Okay, so here you see the RNA in red being made in a 5' to 3' direction. Now, RNA polymerase then has to reach a terminator, and this is another terminator sequence, and this is where both the enzyme and the newly formed nascent RNA strand detach, and then the DNA kind of rewinds and coils itself back up. Okay, so that's why you see the 3 parts of transcription: initiation, elongation, and termination. Now, the other thing that you have to remember is that this RNA that is made is not ready. This RNA is not ready to leave the nucleus, so further post-transcriptional modification needs to happen in order to form a mature RNA. And this is what we're going to look at next. So in this next page here, we focus on the next understanding that states that eukaryotic cells modify mRNA after transcription. So this doesn't really happen in prokaryotic cells, but it does happen in eukaryotic cells. So mRNA modification is known as splicing. So there are 3 post-transcriptional events that must occur in order to form a mature messenger RNA. So the 3 events are known as the capping event, the polyadenylation event, and the splicing event. So we're going to see how each process happens here. So here you have your RNA that has just been newly synthesized from DNA. Now notice that this RNA that is newly made has all of these intervening sequences. So introns are known as intervening sequences, and they were copied from DNA because DNA has a lot of junk DNA, remember that? So not everything needs to be translated into a protein. So the RNA needs to modify the original RNA. In other words, it needs to take out the introns. So all of the introns are spliced with an enzyme known as spliceosome. So that's something that happens in the process as well. Now, something else that happens is known as capping. And in capping, this involves the addition of this methyl group right here. So you add a methyl group to a 5' end, and what this does, it creates what is known as a methylated cap, and it protects the mRNA from degradation from exonucleases. And these are enzymes in the nucleus that degrade parts of a genome, for example. So now it allows this transcript to be recognized by the translational machinery. So in other words, this DNA is ready to be exported out of the nucleus, and it is ready to go to the ribosome and make proteins. The other step that happens is polyadenylation, and this is when in the 3' end of the mRNA transcript, you add a poly A tail. And really all that is a whole bunch of adenines, anywhere from 150 to 150 adenines. And the poly A tail, what it does, it improves the stability of the RNA transcripts, and it also facilitates its export from the nucleus. Remember that this mRNA, again, is made in the nucleus, and it needs to be exported out by the nuclear pores. We already talked about splicing, and that's when all of these introns here were removed. These introns, intervening sequences, or intruding sequences are no longer needed. Exons are express sequence, so these exons do remain. So these exons then need to be joined together, and they form a continuous sequence. Something to keep in mind also is that remember that in mRNA, you're going to need a start codon and a stop codon. And if you remember from HL1, what was the start codon? The start codon was AUG, and I can't write here with my pen, and the stop codons were UGA, UAG, and UAA. Okay, so there you have it. That's how you get the post-transcriptional modification. This mRNA is now ready to leave the nucleus, and it's ready to start translation.